Hi everyone, thank you for coming to my talk this afternoon. My name is Darian Lozon and I am a third-year master's student at West Virginia University. I'm going to be discussing today one of my thesis chapters involving the environmental factors that are related to heavy metal bioaccumulation in freshwater turtles. And as we are familiar with the different land use practices that are found within a watershed, we have roads that allow for ease of transportation for people, as well as agriculture for food and different mining practices for energy. And although these land use practices are beneficial for us, they may be introducing higher quantities of heavy metals into a wetland system that the metals might naturally be there, but they're in much heavier concentrations. And where the wetland could potentially be unhealthy, the plants and animals might also be negatively affected by the presence of these heavy metals in high concentrations. And this is kind of where freshwater turtles come into the conversation, where the turtles are accumulating these metals. Where are they getting these metals from? And for turtles, a lot of where they're getting their metals from is potentially through the bioaccumulation from their prey items. For snapping turtles, for instance, they are known as opportunistic generalists. So they'll practically eat anything that they can get their mouths around. And with larger prey items, particularly, they run into the problem of accumulating more heavy metals in a shorter amount of time because those larger prey items have higher concentrations of heavy metals. What embryonic stages of turtles can run into is the maternal transfer of heavy metals from the mother to the offspring. And along with the maternal transfer, there is also the contaminants in the soil. As they are developing in their nests, they are surrounded by contaminated soil, and the egg that they are developing in is a semi-permeable membrane. So there's water, there's air, there's heavy metals that are going through that semi-permeable membrane as they're developing. And the problem that these embryos can run into as they're slowly accumulating these heavy metals is if it reaches a certain point, they could experience birth defects as well as also mortality if it reaches high enough. And we have the answer of, okay, we have the idea of where these heavy metals are coming from, but how much are they actually accumulating? And non-destructive tissue sampling is where that comes into play, with comparison to different sort of methodology, such as more invasive type of methods that involve muscle tissue or liver tissue. These non-destructive tissues, such as blood or claws, or you could also call them toenails, they are comparable to those more invasive methods. And blood, interestingly, has a more acute concentration, so that's more short-term, immediately, more along the lines of what they ate that week kind of idea. And with claws, they have a more long-term chronic accumulation time stamp, if you will. And with this type of non-destructive tissue sampling, we're able to have these type of studies that are temporal. So we can answer the question of how many milligrams per kilogram of lead, for instance, can a turtle accumulate in a year if you're able to test or sample the same turtle for multiple years. And where my study comes into play is, because this is a pilot study, I just wanted to capture turtles, collect tissue samples, and get them analyzed to see not only what heavy metals are in these turtles, but what the concentrations are. And once those concentrations were determined, we were able to see how these different ideas are sort of related. How does land use potentially relate to turtle tissue or wetland soil? And we could go one step further and say, okay, this turtle is interacting with its environment. How is the wetland soil that it's living in possibly related to the concentrations that are found in its tissue? And the study was conducted in north central West Virginia in Preston County in the Upper Deckers Creek watershed. And this is a really interesting area because you can notice that it once was contiguous deciduous forest, but there's a noticeable belt that goes straight through the middle that is consistent of agriculture, whether it's cultivated crops, hay, or pasture land. And there's a little segment of mine land in the north central region of the watershed, which is really interesting to me, and we'll talk a little bit about that further along in the talk. But the study sites that I conducted my research in, I had 29 sites throughout the Upper Deckers Creek watershed, 26 of which were your typical ponds or lakes, and 3 of them were rivers. And they ranged in size. So the smallest wetland that I surveyed was 0.04 hectares, while the largest was 8.41 hectares. And that'll be important as we go further along as well. But once turtles were captured at each of these sites, I collected 1 milliliter of blood from each of the turtles from the caudal vein, which is just a straight vein that goes through to the end of the tail, as well as 1 to 2 millimeters of toenails. And the number of toenails really varied based on the species as well as the quality of the toenails at the time. And all of the tissue samples prior to analysis were stored in -20 degrees Celsius. And both the tissue samples and the soil samples that I'll talk about in a second were analyzed using an inductively coupled plasma mass spectrometer. Talking about the soil, we collected 2 soil samples from each of the wetlands, so we were able to get a little bit of an average of concentrations that are found at each of the wetlands. And the samples were refrigerated until analysis and analyzed the same way as the tissues, as I mentioned previously. And the size of the wetland comes into play when it comes to land use analysis. So we wanted to look at the type of land use practices that were surrounding these wetlands, but we wanted to make sure that the size of the wetland didn't skew how much of land use practice was present. So we calculated a percent composition with 4 different buffer sizes. We had 1 kilometer, 500, 250, and 30 meters around each wetland, and that's what we used for our analysis. And for our analysis, we conducted a redundancy analysis for both the turtle tissues and the wetland soil. So redundancy analysis simply answers the question, how are these different things related to one another? And it'll make a little more sense once we look at the actual plots themselves. But for turtle tissues, we wanted to take into account of covariates that were related to the turtles, such as species and size. So we used straight line carapace length for that to account for the size classes of our turtles. And we also had geographic sort of covariates. So we were looking at not only percent of agriculture and forest land use, but we also wanted to look at proximity. So for proximity, that's in meters, and we wanted to see how far away these wetlands were from roads and mine land. And if you recall, it's just that one section of the north central portion of the watershed that's really the only mine land that's present in that area. And similarly, we did for wetland soil, we looked at how the different land use practices in proximity were related to the concentrations that were found. And for each turtle tissue or wetland soil, we did a different redundancy analysis for each of the 4 buffer sizes. And to give you an idea of the size of my samples, I collected a total of 78 for blood samples and 62 for nail samples for each of the species. And with that said, the turtle heavy metals for blood, it wasn't too surprising that a lot of the concentrations were close to 0. As I mentioned before, the blood concentrations are more acute. It's what the turtle ate that week, more short-term accumulation. And with cadmium and chromium, for instance, a lot of the samples were below the level of detection. And for those samples, we just assumed that it was 0, that it was too low of a concentration for the mass spectrometer to even measure it. And kind of on the opposite side of the spectrum, zinc in nail samples was significantly greater than any of the other concentrations of heavy metals in either of the tissue types. And I'm not entirely surprised about that or really concerned about that, just because it is commonly found in the literature for high zinc concentrations to be found. And that could potentially be due to, even though these guys are omnivorous turtles, they do have a protein-rich diet. So that could potentially be related to their zinc concentrations. So this is the RDA, and for each vector, they are determining direction as well as magnitude. So if an arrow is really long, pointing toward one of the heavy metals that was looked at in the turtles, that would mean a positive, strong correlation or relationship. So for instance, soil selenium and turtle tissue selenium, there is a strong positive relationship there. On the other hand, we have a strong negative relationship with mineland and carapace length. And there seems to be an equal opposite relationship between 5 out of the 6 heavy metals and the 2 different species, with snapping turtles having a positive relationship with those heavy metals. Looking here at the soil, I didn't really have much to be expected. This is more exploratory, seeing what the concentrations were like. The outlier with lead and zinc was really interesting. It was the same site, and I'm not entirely sure what's going on there, but that site in particular had a history of lead contamination. So that could potentially be what's going on there. I'm not entirely sure about the mechanics of soil and how heavy metals are reacting in that sort of environment. So if anybody has a little bit more of a background there, I would love to chat with you a little bit about that. But looking at the RDA, forest is really the primary driver, and both of these RDAs are within the 250 meter buffer. And forest is the main driver, which is really interesting. So regardless of whether it's lead, zinc, or mercury in the soil, the increased amount of forest within that 250 meters of the wetland has a very strong positive relationship. And we're kind of seeing selenium again with a negative, strong relationship with distance from mines. So that will be really important in the long run with management implications, and we will talk about that in a second. But just to summarize and make sure that we were able to check all of our boxes with our objectives, our first objective was just to see what the heavy metal concentrations were like in these turtles and if there was a relationship. And we found that carapace length was negatively correlated with tissue selenium. So there was a relationship where larger turtles had less selenium, which is opposite of what I would expect, because as I mentioned before, larger turtles, they are eating these larger prey items so they could accumulate this selenium faster. But perhaps the turtles are able to excrete it more effectively, other sort of things like that. And snapping turtles, kind of along the same lines, was positively related with most of the heavy metals compared to painted turtles. Looking at the relationship now between land use and the heavy metals in turtles, mineland and percent agriculture was negatively related with tissue selenium. So that selenium is going to be coming into play a lot here on this slide. And land use practices related to heavy metals in soil. We found that forest cover, interestingly, was the primary relationship, the strong positive relationship with mercury, lead, and zinc, which is opposite of what we would expect, but that's what we found. And distance from mineland, again, was negatively related to soil selenium and chromium. So the farther away the mineland was, the less selenium and chromium there was. So maybe there's a relationship there that we need to look further into. And answering the question of whether there's a relationship between the soil in the wetland and the turtles that are living in that wetland, soil selenium and soil mercury had a very strong positive relationship with what was found in the turtles, which was really interesting. And with all of this information, we can gather a little bit of criteria and say, let's make a sensitivity analysis for where we can put created wetlands where they could be the healthiest per se. And answering the question of how far away we should manage these wetlands with a riparian buffer of some kind. Interestingly, both of my RDAs were the best run within that 250 meter buffer. And that's not to say that 250 meters is the end all be all number, because not all wetlands are the same and they shouldn't be managed that way. And Interestingly, mine land kept coming up, and selenium and chromium are two of the primary pollutants that are related to coal mining, which is fairly common in West Virginia. So that one north central blip in the watershed could potentially be having a larger effect on wetlands in this particular watershed that we might not know about. And with that, I want to thank all of the people who helped to make this happen, because I definitely could not have done it myself. I want to give a shout out to the Society of Wetland Scientists and Dr. Dave Samuel and his family for funding this project in many different ways, as well as West Virginia University, NSF, and the NRCCE for analyzing all of our samples, and all of my interns and volunteers, because I definitely could not have done this study without them. And with that, I want to let you know that my Q&A office hour is on October 2nd at 2 p.m. And if you have any questions, I look forward to chatting about turtles with you. Thanks.